Yes. So, what is CRISPR-Cas technology? So, this is uh, adaptive immune mechanism of bacteria or archaea, mainly prokaryotes, to defend any foreign viruses or phage particles. Our researchers identified that bacteria or archaea has this CRISPR-Cas defense mechanism, which is used to uh, defend phage or other infections. For example, any virus or phage, bacteriophage particle infects bacteria, then the bacteria has the CRISPR gene elements. So CRISPR is termed clustered regularly interspaced short palindromic repeats. So these are genomic sequences having clustered palindromic repeats and some spacer sequences in between those clustered palindromic repeats. So these spacer sequences will imbibe any foreign phage DNA, and then it acts as a memory or adaptive immune memory. And when the bacteria is reinfested or reinfected with the same virion particle, then the bacteria will activate this CRISPR-Cas immune mechanism, and it will cleave or it will degrade the foreign DNA, so that the foreign DNA is no longer active to infect the bacteria. So, in a broad term, this is a kind of immune mechanism present in bacteria and prokaryotes, and the researchers have did significant uh, breakthroughs, and they have re-engineered this CRISPR-Cas mechanism to use as a gene editing tool for the human therapeutics or diagnostics or other applications. So this is actually a native adaptive system of bacteria, which scientists have re-engineered to use as a gene editing mechanism.